So my name is Nur Ashikin. Next, I will further elaborate on the structure of the two types of protein that are used in existing on design of drugs for treatment of disease. So first protein is neuraminidase. So neuraminidase is a type of protein, or also called as Na, that is a tetramer of four identical polypeptides that exist on variant surface. And the four monomers fall into four distinct structural domains that consist of head, stalk, transmembrane, and also cytoplasmic tail. So this is the structure of the neuraminidase. So we go through first to the structure of the Na itself. So the catalytic head of Na consists of a box-shaped structure with four monomers, and each monomer is in the form of a six-bladed propeller structure, with each blade having four antiparallel beta sheets that are stabilized by disulfide bond and connected by loops. So a functional catalytic site is present on the surface of each monomer, and it arranged sideward rather than upward. This is to promote the ability to cleave the sialic acids from nearby membrane glycoprotein to prevent the virus trapping. So the active site is located in a deep central cavity in each of the four monomers that consist the active form of the neuraminidase. The active site is so important for virus spreading and is highly conserved, making it desirable target for neuraminidase inhibitors that can be used in antiviral therapies. So next is the transmembrane domain, which the N-terminal hydrophobic transmembrane domain attaches the Na to the viral envelope. So this attachment provides the signal for translocation from the endoplasmic reticulum to the apical surface, as well as the association with the lipid raft. So actually, both the cytoplasmic tail domain and the transmembrane domain contribute to the transport of the glycoprotein to the apical plasma membrane. So further with the stalk domain, so the cysteine residues at the neuraminidase stalk may assist with the tetramer stabilization by enabling disulfide bond to form between each monomeric Na unit. And the presence of the carbohydrate side chain within the stalk is thought to contribute further to the stability of the tetramer. So basically, the length of the stalk region can have a significant impact on particular virus characteristic. So previous study has proved that reduced stalk length will impact the Na activity of the variants, because the reduced height may hinder access to cellular sialic acid expressing receptors, and that the towering HA blocks the shorter Na catalytic domain from gaining access to the sialic acid. So the lack of the Na function would trap the viral particle and prevent its attachment to the cell surface receptor for viral entry. So reduced stalk size may also alter the geometry and dynamics of the enzymatic pocket itself, modifying its affinity for sialic acid as shown in this figure on the next slide. So this figure shows the part A, the optimal Na stalk length enable the Na catalytic head access to the sialic acid expressing receptor, while for the figure in B shows that the reduced stalk length may impact the ability of the Na to contact sialic acid on the cellular receptor, as the neighboring HA may sterically hinder the shorter neuraminidase catalytic domain from gaining access to the sialic acid. So done with the neuraminidase protein. So we next, I continue with the second type of protein, which is cyclic peptides, or to be specific, we have discussed on cyclic P7 peptide in the next few slides. So this is the figure that shows the structure of the cyclic P7 peptide. So generally, the cyclic peptides are usually composed of 5 to 14 amino acids and have a molecular weight of about 500 to 2000 Dalton. So the moderate size and diverse functional groups of
peptides ensure that the contact area is large enough to provide high selectivity and their potential to form multiple hydrogen bonds that can lead to strong binding affinity. So in addition, the cyclization of the peptides also facilitate the passage through the cell membrane, thus broadening the potential use of cyclic peptides in generating the structure and also the function features that can be used as the pharmaceutical agent. So because of that favorable characteristic features of that cyclic peptide, various cyclic peptides have been exploited for biomedical and other purposes that we'll be discussing more in the next few slides.